Hello, I'm Dana Hart. My specialty is in neurogenetics with an emphasis on the neurotransmitter oxytocin. I have another article to talk about today. With neurotransmitters, there are two proteins that must be coded for genetically: the oxytocin itself and the receptor for oxytocin. These genes are found on two different chromosomes in humans. My special interest over my studies has been a gene that codes for the oxytocin receptor. In that chain of DNA, a particular position of the code has mutated. In genetics, we call this a SNP, which is just the letters S and P. A SNP is a single nucleotide polymorphism. What that means is at this particular point in the DNA, people can have either one nucleotide or a different one. You may remember from basic genetics that the DNA code is made of four letters: G, A, T, and C. The SNP that I've taken a special interest in is called RS53576. That code tells us exactly where the mutation is on the genetic code. The possible letters in this position are going to be either G or A. It is thought that the ancestral copy of this gene had the G at that position, and that a mutation arose where G was switched into an A. I've done quite a bit of research trying to confirm this. I have an article here today which illustrates one of the methods used in figuring out which gene came first. In this article, the frequency of each mutation is mapped to populations around the world. The G version of the gene is very common in Africa, which is thought to be the original home of humans. In contrast, the A allele is overwhelmingly common in Asia. In future videos, I will start to explain some of the effects that RS53576 appears to have on people, and how our personalities and even culture seem to differ based on which copy of the gene we have. For now, I just wanted to answer a common question people seem to have about it, which is which version of this gene came first. From my research, it seems that the G, or guanine, was in the position in this long string of letters that make up the oxytocin receptor, and a mutation event occurred that caused the polymorphism later on, introducing the version of the oxytocin receptor which has an A, or adenine, in that position. The article I'm using as a reference today has a large table comparing gene frequencies for many populations. For example, in the United States where I live, it was listed that the G allele was found in about 60% of the DNA strands tested. That's all I'm going to discuss about this particular thing today, but it will go on. This is just the beginning. Thank you for tuning in. I appreciate you, and please watch for more videos in the future. Goodbye.